right? Now, what I'm going to do now, I am going to start, I'm going to start with talking about chapter 25. We did finish 24. Now, 25 is a natural connection with 24, right? 25 is all about the actual, 25 is about what happens once the food gets into your system. So 24 is how do we bring food into the body? How do we bring it into the cardiovascular system? Important as hell. But when we talk about 25 is what do we do with that food once we bring it in, right? Once we bring that food in, how do we bring that food and all that nutrients? What do we do with it in terms of how do we make ATP out of it? As well, how do we then store it for the future? All right. The overtelling sign, or the one thing I want to always have you keep in the back of your mind is that as we evolved, food was never always present like we see now. So when there's a time of plenty, when you're eating and there's a lot of food, you want to break everything down and store it for the future. So then when there is no food available, you have lots of fat and a lot of glycogen, hopefully, right? So again, the main purpose is to make sure that we have enough food for the future. And what we're gonna do in terms of the nutrition is kind of discuss, right? How does all of the things we eat, how do we use it for energy? And how do we store it for times when there is no food available? Now, usually when I say right after we have eaten, that's a time of plenty. It's a time of feasting. You've just eaten. The whole purpose at that point is to receive all the nutrients, bring it into your cells, and some cells will use those nutrients and start producing ATP. Some cells will store it, like the liver. The liver is not very important as we are digesting, as absorption is occurring. Not very important. All it does is produce bile. The liver is important when we have absorbed the nutrients or when there are no nutrients available. That's when it's important. It's more important for metabolism than for digestion and absorption. So let's talk about one of the first functions it does. And then it makes sense why the hepatic portal vein is so important and why in just about every lab practical, I have the hepatic portal vein as part of your word list, all right? So one of the first things it does is this. You absorb all three types of monosaccharides. You've absorbed glucose, galactose, and fructose. So that gets absorbed, right? Now, fructose is found as part of sucrose, which is that sweet, sugary, right, material that make fruits so sweet and so good tasting. Well, that fructose is going to be produced when you break down sucrose. Sucrose is also known as table sugar. Sucrose is glucose bonded with fructose. And again, our receptors in our GI tract, in our simple columnar cells with microvilli, those cells have lots of plasma membrane surface area because of the microvilli. Because you have a lot more volume and space for the plasma membrane, we also have a lot of space to hold receptors. So what we get then are lots of glucose, lots of fructose, and lots of galactose receptors. Now, go back to recordings that I'm in the process of posting for the end of chapter 24. And we talk about, how we absorb glucose, galactose, and fructose. It's absorbed by what we call secondary active transport, where the sodium potassium pump on a side that is opposite of where the food is coming in. So you have a sodium potassium pump. It doesn't drive sodium potassium, and that's the main purpose. No, the main purpose of this sodium potassium pump is to produce a low sodium environment inside each and every cell that makes up your small intestine. So each and every billions and billions of cells in your small intestines, all those simple columnar cells are going to have a low sodium environment because of the sodium-potassium pump. And what happens then, when sodium and glucose are found in the lumen of your small intestine, you've broken down your carbohydrates. Now you have sodium, you're going to have glucose, you're going to have fructose, and you're going to have galactose. What happens? Well, what happens is, sodium wants to come into the cell because there's low sodium inside the cell. Sodium comes in through sodium receptors that are also going to be bringing in glucose. So it's a combination sodium glucose receptors, sodium fructose receptors, sodium galactose receptors. So as soon as sodium goes in, glucose goes along with it. This is the reason why Gatorade works so well. Why does Gatorade, Powerade, Pedialyte work? Is because it's got a little bit of, well, for Gatorade, a lot of sugar, right? A lot of glucose and fructose. It's got a lot of sucrose in it. And what happens is, when you have a lot of those sugars, and now we kind of mentioned that Gatorade, Pedialyte, all of those have a lot of sodium. As sodium goes through those receptors found in the simple columnar cells, it brings in glucose. Now you have sodium and glucose co-transported in. What happens? Water follows, right? So the more of the actual solutes that go through, the more water follows. Perfect, that's what Gatorade is supposed to do, right? Enhance water reabsorption or absorption. So by bringing 2 salts and 2 solutes through, you have more of an osmotic push. Perfect. Now, once we bring glucose, galactose, and fructose into and past the simple columnar cells, we need to bring it in and for some reason, my mic went crazy. All right, so hopefully we didn't just get, worked on by some of these people here, right? Then we didn't just get Zoom bombed, but if that was it, that's fine. All right, so when we started talking about let me go back here, right? The processing and then the absorption of the carbohydrates, you will absorb glucose, galactose, and fructose. We will absorb all of it. But what we're going to see is that once we absorb all of it, we are also going to then convert it into glucose. So all the fructose, all the galactose goes through the hepatic portal vein. Eventually, the hepatic portal vein allows you to bring all those nutrients to the liver first. Remember, there's a reason why I want you guys to know that the superior mesenteric vein, the inferior mesenteric vein, as well as the splenic vein, all fuse together to form the hepatic portal vein. Now with the superior and inferior mesenteric veins, well, that's going to be carrying a lot of nutrients, right? All of your amino acids, all of your carbohydrates that you've just absorbed goes through the superior and inferior mesenteric veins, through which then kind of fuse to form the hepatic portal vein, and then the liver receives those carbohydrates immediately. And all of the fructose and galactose gets converted to glucose. Now, the great thing is this. Now, the last thing we want to do is have the liver make use of glucose as a fuel source. Remember, the liver, we're going to see, will be the main catalyst organ, so then you can store glycogen, right? So in order to do that, it doesn't make sense for the organ that should be storing glucose to also be using it. Why? It might be trying to steal the glucose for itself. So what we're going to see is the liver itself does not use glucose as a fuel source. it uses alternative fuels, including fatty acids and ketones. So it doesn't use glucose, and it's perfect because you don't want it to. The last thing we want to do is have the actual liver that's supposed to be storing all the glucose to use that glucose. It will try to steal it for itself. So what happens with the liver is it receives all those 3 types of monosaccharides, converts it to glucose, all of it, now it's glucose, and then the liver turns the glucose into glycogen. Remember, glycogen is how we store glucose. And we only store glucose in 2 areas, skeletal muscles and the liver. So the weird thing is this, we can store it in the skeletal muscles, but the stored glycogen in skeletal muscles will only be stored for that skeletal muscle to use. In other words, it does not let them, the skeletal muscle does not release any of the glycogen that's broken down to glucose. It doesn't release the glucose into the bloodstream. It doesn't share the glucose that it has stored. It only uses it for itself. So the only area where we can store glycogen, and then when we are fasting, Ramadan just started. So a whole, couple billion people in the world have started this fasting state. Well, what happens is in the fasting state, your blood glucose is going to drop. So the liver then needs to take that glycogen it stores, break it down, and then release the glucose into the bloodstream for the brain to use. The brain can only use glucose as a fuel source. Most other organs can use alternative fuel sources, but not the brain. So it's important for the liver cells then to have a big supply of glycogen. So then when there is a time of fasting, well, what happens? Well, the liver can then take that stored glycogen, break it down completely, and then turn it into glucose. The glucose then goes into the bloodstream for hopefully the brain to use. By the way, you guys have seen those old Snicker commercials where the people get really moody and grouchy and, kind of being a pain in the butt when they get hungry. The reason why is simple, right? The brain is solely dependent on glucose. When you haven't eaten in a while and you're hungry, what happens? Your blood glucose is starting to drop. The first organ that feels it, that senses it, and starts having a dysfunction is going to be your brain. So in some people, they just get tired and lethargic. Some people, they get moody, grouchy, right? Angry, but if people call it hangry. So my son's like that. He gets really angry. We don't know why sometimes. And then it's because he's hungry. So that does happen to some people. Now, what other kind of nutrients we have? We have fats. Again, with fats, lipids, 95% of the fats are in the form of triglycerides. Triglycerides are used for energy as well as stored in adipose tissue and liver. There's a limit to how much glycogen we can store. The liver can only store so much of it. There's almost no limit to how much fat we store. If you've ever seen my 600-pound life, you can notice that there's almost no limit, right? Now, again, the greater amount of fat, the more time we can have as we have starvation occurring, right? So the greater amount of fat, the more energy we can store for times when we are starving. So we have saturated fats, unsaturated fats, cholesterol, phospholipids, linoleic acids. These are all the different types of fats, right? Uses of fats include triglycerides. The fats that we see in adipose cells, those are triglyceride. They serve no other purpose than to be broken down and to produce energy. This is why our body likes to store it, is because unlike glucose, glucose can be converted to, right, marker proteins like your ABO blood groups, right? It can be used like that. So it can be converted to, certain other types of sugars, right? So there's another purpose for some of the monosaccharides we ingest besides energy. But for triglycerides, there is no other purpose. The sole purpose is to store in times of plenty and then break down in times of fasting or starvation. So that's a big difference between glucose as well as other fuel sources compared to lipids. Now, triglycerides are the big difference right here, used to produce ATP only. We see cholesterol can be eaten or manufactured in the body, and cholesterol is important because it's going to be, right, it can be eaten or manufactured, and there are a lot of different things that cholesterol can become, right? So in terms of cholesterol, our body produces native cholesterol. So even people that have high cholesterol, and when they have high cholesterol, their cholesterol levels are incredibly high. And when you look at their diet, they don't eat a lot of foods with cholesterol. So people are wondering, why do we have this high cholesterol count then? Well, it's because your body produces about 65 to 70% of the cholesterol needed for its reactions. Cholesterol is required to make bile. Cholesterol allows you to, right, kind of solidify the plasma membranes. Cholesterol is required to produce testosterone and estrogen, which is a huge, right, kind of function of cholesterol. It is the backbone for steroids that we use in our body. So that's important use of lipids other than for kind of storage for energy. The only one that's stored just for energy is triglycerides. Every other one, they have different functions. Eicosanoids derived from fatty acids involved in inflammation, blood clotting, tissue repair, and smooth muscle contraction. We talked about phospholipids already, so we're not going to go into too much more detail about that. In terms of metabolism, keep in mind, our bodies are meant so then we can store. So when we say metabolism, metabolism is the catabolism, which is the breakdown of larger molecules, and anabolism added, or you can even say, right, subtracted together. So when all of your anabolic production or metabolic, anabolic needs, like anabolic steroids, you have something small, you make it to something bigger. Example, glucose, added with other glucose to form glycogen. That is an anabolic reaction, right? Usually anabolic reactions occur when you have a lot of food around, a lot of nutrients in your body. So we like to build when there's a time of plenty. Catabolism is a breakdown, right? So now you have all the nutrients that are already all built up, right? We have increased amount of stores of those nutrients. Now we're fasting because we're Muslims and it's Ramadan. What do we do then? We're going to break down all those macromolecules. So then glucose can be freed from glycogen. It can be released into the bloodstream. And now you can break down fats, break down, right? Usually we break down fats first. So we break down the fats, the triglycerides, and turn it into alternative sources like keto acids for the rest of the body to use. Because keep in mind, the last thing we want to do is break down our stores of glycogen, release the glucose, and then have other organs steal that glucose that should be going for the brain. So metabolism is a combination of all the buildup reactions subtracted by all the breakdown reactions. Normally, before the last 100 years, or actually I'd say the last 60 years, right? We usually have more breakdown reactions. So we don't build as much. People were thinner, less obese. In the last 50 years, we know we can get food just about any time. And the food we can get at any time are usually full of carbohydrates and fats. In other words, full of nutrients and fats, right? And what happens then is we have anabolic reactions where we build more and more and more. At the same time, we don't have those spans of time where we're fasting. In the old days, right, you'd have a big meal and then you might not eat again another big meal for a day, maybe a day and a half, sometimes even 2 days. So then when you have that big meal, you want to have lots of anabolism. So then you can store everything for those 1 or 2 days where you're not eating and you're breaking down, right? Usually the catabolic reactions occur for a much longer period of time. So when you have more breakdown than buildup, you're not going to gain weight, right? We say you have a high metabolism then, right? Nowadays, we think a high metabolism is good, right? Because now you can break down, you can eat all you want, yet not gain weight. Now, that's only good when there's a glut of food. That would be terrible if you are starving, right? Or there is a famine. If there's a famine and you can't receive nutrients, the last thing we want is a high metabolism. That means whatever you eat will be broken down immediately. You can never store, and because of that, you never store fats, never store glycogen. You're not going to live long like that, right? Now, again, with carbohydrate metabolism, during digestions, polysaccharides and disaccharides are converted to 3 monosaccharides: glucose, galactose, and fructose. Those are all absorbed through the capillaries on those finger-like extensions called the villi, then transported to the liver by the hepatic portal vein. The liver converts much of the fructose and almost all the galactose to glucose. Carbohydrate metabolism is primarily concerned with glucose metabolism. So that could be a good test question: is true or false, right? Only glucose gets absorbed through the GI tract. It doesn't, right? Because fructose and galactose also gets absorbed. But most, if not all of it, goes through the hepatic portal vein and gets converted to glucose. That's why you don't hear much of a blood galactose count or a blood fructose count, right? So just review this in terms of carbohydrates. I'm just going to underline some of it. In the GI tract, figure out what happens. Then in the liver, right? The liver convert things like fructose and galactose into glucose. And then because the liver does not use glucose as a fuel source, is going to undergo a process called glycogenesis to form glycogen. Genesis, like when we look at the Bible, Genesis is the building story, right? Same thing. Anytime you hear the word Genesis in its name, it is the formation. So glycogen, genesis, glycogenesis is the formation of glycogen from glucose. Now, some of the cells, right? So almost all of the glucose, though, stays in the bloodstream, as the glucose stays in the bloodstream after the hepatic portal vein. So the liver receives first dibs of the glucose. It receives galactose and fructose and changes it into glucose. It stores the glucose as glycogen, but a lot of the glucose will then go through the bloodstream still for the other cells to use, for the brain to use. So as the glucose goes to the bloodstream, your cells of the body can oxidize it to form energy. So that's where the Krebs cycle, that's where glycolysis comes in, right? So in terms of the oxidation right here, this is the oxidation of glucose. Whoops, butchered that. Oxidation of glucose via glycolysis and the Krebs cycle. Right? Now the Krebs cycle is also called the citric acid cycle, and we'll talk about that later on. So keep in mind, once it goes into the cardiovascular system, the liver gets first dibs, but it doesn't use all of the glucose. Keep that in mind, right? Most of the glucose bypasses the liver, and then it goes into your bloodstream for the other cells of your body to use. When that happens, then the glucose can be oxidized by those cells to form lots of ATP, especially if there's oxygen. So we'll talk about the fate of glucose, right? Glucose is the body's preferred source of fuel. And the fate of absorbed glucose depends on the energy needs of the body. If the cells require immediate energy, glucose is used by all the cells and oxidized immediately. Glucose, though, can be used to form amino acids, which then can be incorporated into proteins. Excess glucose can be stored by the liver and the skeletal muscle as glycogen. Process we call glyco, and then highlighted the genesis. Genesis is formation. If glycogen storage areas are filled up, liver cells, fat cells can convert glycerol, glucose to glycerol and fatty acids to form triglycerides in a process we call lipogenesis. So we will need to know about the lipogenesis, and we have lots more slides later on to discuss it. In other words, if you already use all the glucose for the energy needs you need for that time frame, you have an excess of glucose. So what happens? The liver will then store as much glycogen as it can, but it can only store so much. Once that's filled up, then if you still have glucose, we convert it into triglycerides, right? Again, genesis, lipogenesis. Lipo for lipids, genesis for formation. Whenever you hear the word genesis, it is the formation of that stored macromolecule. Here for lipids, right, which is triglycerides. Now, kind of talk that right, in terms of movement into the cells, when we absorb it in the GI tract, it is accomplished by secondary active transport. Let me see if I have a picture of it. No, right? So in the GI tract, how we are able to absorb it is going to be slightly different to how we absorb it when it's already in our bloodstream and has to go into our individual cells. So in order to absorb it from our diet, as it goes through your small intestine, in order to absorb it, we need sodium, glucose, symporters found in the small intestine. Right? Now, we have a different set of receptors when the glucose is already absorbed and now it's in the bloodstream. As it goes into the bloodstream, most other cells of our body, except the liver, right? Most other cells prefer to use glucose if available, if there's a lot of it. Now, what happens is those other cells of the body will then allow glucose to go from the bloodstream into those cells. So when you eat a high protein, high starch diet, meat and potatoes, right? What happens? Well, when you eat the potatoes, potatoes are starches, which is just like glycogen. So you break it down into individual glucose molecules, you absorb it, and then you transport it into the bloodstream. As it goes into the bloodstream, what happens to your blood glucose levels now? It's going to elevate, right? Your blood glucose is going to go high. It's going to be very high because you just absorb all the glucose. As the blood glucose is elevated, it triggers the release of insulin. Insulin allows us to insert a second group of glucose receptors called GLUT receptors, right? The GLUT receptors then are placed on the plasma membrane of all of your cells of your body, right, except the liver. So these GLUT receptors are very important. They're the ones that get actively produced in the presence of insulin. So when there is no insulin, there's a lack of GLUT receptors. What happens? Well, if there's no GLUT receptors, your blood sugar is high, blood glucose is high, and now it can't go into your cells. Because it can't go into your cells and out of the bloodstream, it stays in the bloodstream. As it stays in the bloodstream, your blood glucose gets to elevate and it gets higher and higher and higher, all because of a lack of insulin. This is why we see massively high blood glucose levels with type 1 diabetics. Type 1 diabetics, they really, really have a hard time, right? Because they have no insulin at all being produced. When that happens, They have no insertion of GLUT receptors, and the cells, they should be receiving glucose because you've just eaten, but because of the lack of insulin, they're not. So those cells are actually starving because it's not receiving the glucose. So it causes, all right, more of a freak out, right, which then causes the liver to release more glucose. And now you have this bad positive feedback, right? This is why you see such astronomical numbers for blood glucose in type 1 diabetics. Type 1 diabetics that forget to take their insulin, I have seen in my previous life as a doctor, I've seen patients with blood glucose levels that were 6, 7, 800 milligrams per deciliter of blood. I mean, that's like 10 times the normal levels, right? Why? It's because of the lack of insulin, the glucose that should be going into the cells doesn't. The cells, all the cells of the body freaks out, causing more release of glucose from the liver. Now you have this huge high spike of glucose from eating, and now you have a secondary spike because the liver now is releasing more glucose. That's where we get the huge numbers. Now, those GLUT receptors, probably easier to see what happens. All right, GLUT receptors right here are going to be placed into the plasma membrane. As it plays into plasma membrane, insulin increases the insertion and formation of GLUT receptors. More GLUT receptors from insulin, more channels for glucose to go into the cells. As glucose goes into the cells, though, what happens is, as glucose goes into your cells, right? Before, your cells have low glucose levels, high glucose levels in your bloodstream. There's a glucose concentration gradient. The glucose receptors only facilitates the movement of glucose from high concentration in the bloodstream. It goes through those GLUT receptors into the cells where there's low concentration. Perfect. So what happens then? Well, if you have a lot of the glucose going into your cells, you have billions of cells in the body, trillions of them, all receiving the glucose molecules now. Well, what happens then is your blood glucose starts to really drop. And now we're going to see you're going to have high amounts of glucose in the cells and lower amount in the bloodstream. So we should have the reverse movement of glucose now from cells to the bloodstream. And that would not be good. So we don't want that to happen. So what we need to do is trap the glucose once it goes into the cells. This is why the first step in glycolysis is so important. As glucose goes through the GLUT receptors, it gets activated, right, by an enzyme that allows us to produce glucose 6-phosphate. By producing glucose 6-phosphate right here, you now have changed glucose. It's no longer just glucose. Because of that, the glucose is hidden inside the cell. So then you don't have this huge amount of glucose coming in. Now you have high glucose concentration in the cells, low outside the cells in the bloodstream. And now you don't have, the glucose wants to go out now. By changing it into glucose 6-phosphate, you basically hide it. And you make sure that the glucose, the normal glucose levels, aren't so high inside the cell. Why? Because it's no longer glucose, it's glucose 6-phosphate. By putting that phosphate group in glucose, you are trapping it, making sure that it doesn't leak back out. Now, the other good news is this. By producing glucose 6-phosphate, now we can actively run glycolysis. So not only is it a trapping step for the glucose, so then the glucose doesn't exit out, it also starts the oxidation of glucose. And we're going to see, right, when we need that energy right away, we're going to take glucose, turn it into 2 pyruvates in the cytoplasm. The 2 pyruvates goes into the mitochondria. In the process, it gets turned into 2 acetylcoenzyme A's. Then it goes and becomes a substrate for the Krebs cycle. So then we can produce a lot of ATPs later on. In other words, formation of glucose 6-phosphate starts the process of taking energy from the glucose. Keep in mind, in terms of glucose, the energy is in the bonds between all the different elements, right? The other elements, all the carbons that we see, the oxygens that we see, the hydrogens that we see in glucose, all of those elements, what happens to them? It turns into carbon dioxide and water, are waste objects at the end, right? So it's not the elements that are important, it's the bonds between, right, those elements that's important. That's where the energy is. So when you break apart the bond between carbon and another carbon, carbon and another oxygen, when you break apart the bond, you are giving away energy. The electrons are then transferred, right? The elements that's left over, that just becomes waste. It's the bonds between the elements that are important. So in terms of glucose oxidation, right, we're gonna see that in terms of like the steps, right, the steps of glycolysis, the steps of the Krebs cycle, this, right, that's going to be in a quiz. That way it's, you know, you guys have your notes in front of you, right? I don't want to make sure that it becomes overly burdened that you memorize all the reactions of the Krebs cycle. I want you to know generalities for the test. For the quiz, I will ask you specific reactions. But the good news is the quiz will be probably about 15 questions, and it's going to be specific on the Krebs cycle, electron transport a little bit, and glycolysis. You will have your notes in front of you though, right? So the first step in producing energy from glucose is taking that glucose and turning it into 2 identical 3-carbon pyruvates. Keep in mind, glucose is 6 carbons, right? Because glucose is 6 carbons, what we're gonna see is that we want to be able to split glucose in half. And now you have, all right, 3 carbons over here. So here's our 6-carbon glucose. 1, 2, 3, 4, 5, and 6, right? Actually, it should be 1, 2, 3, 4, 5, and 6, right? Those 6 carbons will need to be divided in half completely to produce 2 identical pyruvates. That's the sole function of glycolysis, is somehow making one side right here, 1 3-carbon glucose over here, look identical to the next 3-carbon glucose, right? And now we're gonna have 2 3-carbon pyruvates. So that's the main purpose of glycolysis, is to produce pyruvic acid. 10-step process occurring in the cytosol of the cell. Now again, for the quiz, you need to know the reactions. For the test, you need to know where it happens and the general things that happen, right? Which is, for glycolysis, the sole purpose is to take 1 6-carbon glucose and turn it into 2 3-carbon pyruvates, right? In the process, we're gonna produce very little net ATP. We're gonna see there's gonna be 6 total ATPs, but you have to put 2 in the beginning. So you're only gonna produce 4 ATPs through glycolysis. You produce this, NADH. Now, with these, when you're going through any biochemistry, I'm gonna give you a hint. Anytime you add hydrogens to any chemical, you're gonna add electrons to the chemical. The more you add electrons to the chemical, the more energy you're gonna produce. So here we have 2 NADHs, 2 NAD positive, that's very low energy. In the process of forming pyruvic acid, we're gonna take the 2 low energy NAD positive and then turning it into 2 NADHs, high energy, right, intermediates. So these NADHs our intermediates right here. It's our way of storing the electrons that we need. So then we can use it to form ATP later. We're going to see, right? When we form ATP directly, like we see here, we call it substrate level phosphorylation, meaning we just take the material, ADP, and then we add phosphate to it. The good news is it produces ATP fast. The bad news is it's very inefficient, right? So you produce 1 ATP at a time, not very efficient. What we want to do is produce something that makes ATP more efficiently. Where if you have 1 of this, NAD positive, we're going to see by mitochondrial electron transport, for every NADH we produce, the high energy form, for every 1, we don't produce just 1 ATP. We are eventually going to produce 3 ATPs for it. In other words, there's a 1-to-3 ratio, right? 1 NADH will produce 3 ATPs. NADH then, right, is very important because it produces a lot more ATPs. Now, that's only true if there's oxygen available, right? If there's oxygen available, we can take this NADH and we can make it go through the mitochondrial electron transport chain and eventually produce 3 ATPs each for 1 of the NADHs. So here we're going to form 2 of it, right? 2 NADHs per glucose. That's 6 total ATPs by forming 2 NADHs. So if you think about it, right, we make 6 ATPs total directly by, again, direct formation of ATP, right? We call that substrate level phosphorylation, right? Actually, we form 4 here, and then we have to take 2 out. So net, we only make 2 direct ATPs. But the big thing is we produce 2 NADHs and 2 pyruvates. So the most important product of glycolysis isn't the ATPs you make. It isn't even the NADHs you make. That's important, but not as important as producing 2 of these pyruvates. The pyruvates you form are the most important because that will be used to go into the mitochondria. It will be then added to with be converted to acetyl coenzyme A, and then get added to with oxaloacetate. So then you can cycle the citric acid cycle, right, to produce more ATPs and NADHs. In other words, for our purposes right now, understand this is the most important product of glycolysis. Why? Because it'd be used to form more energy. Now, what happens if there is no oxygen, right? So what happens if there's oxygen, that pyruvate gets formed and it goes into the mitochondria. Perfect, right? As it goes into the mitochondria, the 2 pyruvates or pyruvic acids, right? Those words are interchangeable. The 2 pyruvates goes in the mitochondria, gets converted into, right? Gets converted to acetyl coenzyme A, right? Then it helps run the citric acid cycle. So you're going to run 1 pyruvate is going to run the citric acid cycle 1 time. So with 2 pyruvic acids or pyruvates, you're able to run the citric acid cycle twice. And if we're going to see, eventually that's going to produce 30 ATPs. That's a lot. So that's going to be where a vast majority of the energy comes from, is bringing pyruvate in the presence of oxygen, bringing that pyruvate from the cytosol into the mitochondria matrix. Now, this is also the reason why we are going to die if there's no oxygen. If there's no oxygen, we block this aerobic pathway. Pyruvate can't enter the mitochondria. It stays outside in the cytosol. and it undergoes anaerobic pathway, right? In the process, you form lactic acid, right? Now, the worst part about it is this, right? Remember, I said forming the NADH. Anytime you see the H, it's high energy. Well, when oxygen's available, those two NADHs will produce 6 ATPs. But now there's no oxygen. So what we need to do is somehow reform NAD positive again, right? So what we're going to see is in glycolysis, we form the NADHs. And then because there's no oxygen, those two NADHs we form gets immediately converted back to NAD positive, the less energetic form. In the process, you form lactic acid, something that can cause difficulty, that cause cramps, right? In the skeletal muscles, in the cardiac muscles as well. So we're going to see that, yeah, this is the reason why if there's no oxygen, all mammals die because of this. Why? Because when there's no oxygen, we are solely dependent on glycolysis for ATP. That's it, right? And we saw in glycolysis, you made 4 ATPs total, but you have to put 2 in. So all we get is 2 net ATPs from glycolysis. 2 ATPs is not enough for us to live, right? You just don't have enough energy. When that happens, we will then die from that lack of oxygen. The lack of oxygen, we can't get enough ATPs. We can't drive the reactions in our body. When we can't drive the reactions in our body, the body breaks down because there's no energy and you will pass away because of it, right? So in terms of this, taking this pyruvate and turning it to lactic acid, all we're doing here for anaerobic fermentation, right? Anaerobic pathway. All we're doing here is hopefully producing enough ATP to survive. So then eventually you can get enough oxygen. If you don't, and you're in this low oxygen environment, you're going to pass away from this, right? This is a stopgap. That's it, right? Now, let me go back here, right? If we do go back, let's say, right, you're exercising a lot and you produce, you're exercising real strenuously and then you become anaerobic, like doing sprints really fast, right? Well, when you're doing real sprints, you don't allow oxygen to go into your bloodstream, right? Oxygen then is not used as a fuel source. We cannot use the aerobic pathway. What happens then? We use the anaerobic pathway, less efficient. That's why you can only sprint for such a short time, right? Eventually, if you try to sprint beyond that time, you build up lactic acid that makes you cramp up. Now, that lactic acid will make you stop. Once you stop, you catch your breath. As you catch your breath, the lactic acid can be converted back to pyruvate, right? Your liver will take that excess lactic acid in your bloodstream and convert it back into pyruvate to be used later. So that's a good news, is that when you stop, you get enough oxygen in. Now you're not in this low oxygen or no oxygen environment, right? Now you have a high oxygen environment because you are, catching your breath, or somebody give you, right, some oxygen, 100% oxygen. What happens? Now we can convert it back into this aerobic pathway. The liver takes that excess lactic acid, converts it back to pyruvate. The pyruvate then goes into your cells and gets turned into energy again. Perfect. All dependent on the availability of oxygen. So the fate of pyruvate depends on the availability of oxygen. If there's oxygen available, we send the pyruvate to the mitochondria, right? The inside of the mitochondria, the inner aspect, are where all of the Krebs cycle occurs, right? All the reactions occur. That's called a mitochondrial matrix. Perfect. That's where we want it to go. Now, here's another kind of picture at what happens with Glucose, as it goes through glycolysis, the main purpose, form 2-pyruvic acid. That is the single most important product, are those pyruvic or pyruvic acids, right? Take a look. You only produce 2 net ATPs. That's it from glycolysis. You do produce 2 high energy NADHs, but that's only useful, right? The high energy, right? Hi, Anna. Right, the high energy NADHs is only useful if there's oxygen. If there's oxygen, then it goes into a process that we call the electron transport chain. That electron transport chain is like a way of receiving this intermediate high energy NADH, and then in mitochondrial electron transport is a series of receptors and reactions that produces ATPs by moving electrons from one spot to another to another. So NADHs that we produce from glycolysis in the presence of oxygen enters a mitochondria, and in the presence of oxygen, it runs the electron transport chain, which allows us to produce 3 ATPs per NADH. Right? What else happens with the pyruvates? If there's oxygen available, it gets converted to the pyruvate enters the mitochondria, turns into acetyl coenzyme A, and it helps run the citric acid cycle, where we produce even more NADHs, more ATPs. And then here's a second type of high energy intermediate, right? We call these cofactors, NADH and FADH2. Both of those are the high energy forms of those cofactors, right? Again, whenever you see hydrogen with these cofactors, hydrogen is electron. Electrons is energy. So when you see NADHs, that's the energized form. That's where we're going to eventually, those electrons are going to be moved through the mitochondrial electron transport chain to produce a lot of ATPs, right? The vast majority of the 36 net ATPs, the vast majority is obtained through NADH, FADH2, their movement of electrons through the electron transport chain. Right? By the way, how many of you guys have heard of the electron transport chain? Any guys? Right? Let me just unmute, make sure. Right? Have you guys heard of the electron transport chain before? Answer?
Yes, I've heard of it before.
But have you understood it?
But I don't remember much.
Probably didn't understand it well, right? That's okay. Stephen, I know you're taking biochem, so I'm going to call you out.
Yeah, we're getting to it.
Getting to it.
It's complicated, Dr. Lee.
I did it, but honestly,
can understand it, right?
No, I didn't.
Hopefully, as we go through it this time, it makes sense. Okay.
Okay.
Because it's gonna hopefully as we go through it slowly, it makes sense, and you can use it in your other classes. Right? Because you will see it again. You'll see. I mean, how many? We will definitely see Krebs cycle again in your classes, right?
Krebs
You will see the
cycle.
citric acid cycle and the electron transport chain again. So hopefully you can understand the gist of how it works. So then, as you go into the other classes, they can get. In my biochem, they should be talking about it a lot, but I know that they don't sometimes.
I did, but I didn't understand it much.
Perfect. Well, not perfect, but
I'm being honest.
Right. So we hopefully talk about it and it makes sense then. Right. So we're going to use a couple analogies with the electron transport chain as we get to it. Keep in mind, right? The big thing here is hydrogens equals energy. Right? Why? Because hydrogens equals electrons. And movement of electrons, right? The movement of electrons through the wiring of our house is why, right? Is why lights go on. So we know the movement of electrons give us enough energy for lights to turn on. So movement of electrons in our body will also produce some kind of energy. We're going to take that energy from the electrons and turn it into ATP. Right? Now, if you were in my class last semester, we talked about electrons are energy real early on, right? Same thing here. Electrons are energy. So when electrons move from one place to another, you have an electrical current occurring that will then cause energy, right? Energy movement. We follow very similar rules of physics. The big difference is instead of a copper wire, we have like organic proteins that allows the movement of those electrons. Weird, right? All right, let's take a look at glycolysis. All right, glycolysis, again, is the start of glucose oxidation. Number one, I want you to know, right, for the test is the first reaction. That's it, right? In terms of glycolysis, big picture for the test, one of the first pictures is that first step. And the reason why is that the first step, taking glucose and turning it into glucose 6-phosphate, that first step requires ATP, and it not only is done so then we can undergo glycolysis, meaning it is not solely a step in glycolysis, and that's it, right? What formation of glucose 6-phosphate allows us to do is, number one, trap the glucose inside the cell. Now glucose is now changed to glucose 6-phosphate, and it's no longer glucose. When we change it to glucose 6-phosphate, the concentration of glucose in the cell starts to decrease, right? Perfect. Remember, concentration gradient, right? So for the people that just joined us, when we eat, we absorb the glucose into the bloodstream. Now you have a high glucose concentration in your bloodstream. Now, insulin allows something called GLUT receptors to be inserted to all of your cells of the body. Those GLUT receptors are glucose receptors, right? As they're introduced into every cell in your body, the trillions of cells we have, now glucose from your bloodstream goes inside the cell. Well, what happens? If all the glucose in your bloodstream, a lot of it goes into your cell's glucose concentration is going to be elevated now, right? Much higher than in the blood. So now we switch, right? Before it was high in the blood, low in the cells. Now it's high in the cells, low in the blood. So glucose wants to go out now in the opposite direction, right? So we need to trap it. This is how we trap it. We take the glucose as it enters and turn it right into glucose 6-phosphate. In the process, we don't increase our cellular glucose. Why? Because the cellular glucose gets turned into glucose 6-phosphate, and glucose 6-phosphate now is trapped inside the cell. There's no reverse movement from cells into the bloodstream. Now, we'll also see this is also the first step in glycogenesis. So not only is the phosphorylation of glucose into glucose 6-phosphate, not only is it the first step in glycolysis, it also traps all glucose inside the cell. It also is the first step in glycogenesis, right? So that's what makes it so important. Now what happens, right? Now, what we want to do with glucose is a 6-carbon sugar. What we want to do is, right, if you take a look at this, there's no easy way I can break it down and have these 3 carbons looking the same as these 3 carbons, right? I can't split it in half like this. If I split it in half like this, right here, that means that one side only has 2 carbons. Remember, in chemistry, each point is a carbon. So here's carbon, here's a carbon, here's a carbon, here's a carbon, here's a carbon, and here's that last carbon. So each point is a carbon here. So if I split it like this, split this hexagon in half, that means I only have 2 carbon on one side, and the other side has 4 carbons. That definitely doesn't look the same. So what we need to do is change it. How we change it is take glucose 6-phosphate and turn it into glucose 6-phosphate. Now take a look what happens, right? What if I take this and I split it in half right here? What happens? I have one side chain of, right, of methanol, right, CH2OH right here. One carbon, that's a second carbon. So 1, 2, 3 carbons. Another side, right? One, right, 1, 2, 3 carbons. That looks very similar now, doesn't it? All we have to do is put a phosphate group here, right? Then it looks very similar to what we see here. This, we have to put an oxygen, then it'll be identical then. And then just switch these two around, right? In other words, once we turn it into fructose 6-phosphate, it looks similar on each side. Before, if we cut it in half, completely looks different. Even the number of carbons look different. Now we turn it into fructose 6-phosphate here. Each side, 1, 2, 3, 1, 2, 3, they look similar. We can work with this. So this is why we isomerize it and turn it into from glucose 6-phosphate to fructose 6-phosphate. Does that make more sense now, guys? Right? Now these reactions, I do want you to think that it should make sense, right? Now, what else happens? In order to make the right side look like the left side, we need to add a phosphate group, right? Doesn't make sense now? We have to add a phosphate group, right? So right here, we go from fructose 6-phosphate, we add an extra phosphate group right here, and now we have fructose 1,6-bisphosphate, right? Now, if you take a look, if I cut this right in half, these two this carbon and this one, they're identical, right? This carbon and this, right? This carbon over here, well, it doesn't look identical, but we can work with it, right? Take a look. This carbon has an OH group, and on this one, on the other side, it's got a hydrogen. But you know what? It's not hard to put in oxygen there. These two carbons over here, this one to this one, right? They look very similar. So we're almost done, right? So what we have done is turn this weird looking glucose into something we can split in half and have each half look identical to the other half. Perfect. Remember, the sole purpose of glycolysis, the big purpose, is to produce pyruvate, which are 2, 3 carbon sugars that are identical to each other. Now you can start seeing where these 1, 2, 3 carbon sugar, 1, 2, 3 carbon sugar right here, if we split it in half, this half, right, and the other half are gonna, so this half and this half look very similar already, right? Not identical yet, but pretty similar. That's where we are going to, right, split it. As we split it right there, come back here. As we split it on this side and on this side, remember, they're not identical yet. So we're gonna at first form 2 separate chemicals. So here on that left side, we're gonna form dihydroxyacetone phosphate. Similar to what we produce on the right side, glyceraldehyde 3-phosphate, right? So what happens? This and this look very similar. Why? We have a phosphate group on carbon 1, phosphate group on the carbon 1. Right here, right, we have a carbon and it's got just hydrogens right here. We can convert it to this pretty readily, right? And then this can get converted to that pretty easily as well, right? Now, if you've taken biochemistry, you know that primary, I don't know if you guys remember this, right? But primary alcohols, this is a primary alcohol, right? A primary alcohol has carbons, an OH group, and then the other carbon, other bonds of carbon has hydrogens on it, right? It's primary because it only has 1 carbon attached to it. So a primary alcohol
can easily convert to an aldehyde, right? Actually, primary alcohols can be converted into carboxylic acids as well. I used to teach biochemistry, right? So here, this dihydroxyacetone phosphate easily converts to glyceraldehyde 3-phosphate. So what have we done now, right? By cleaving that fructose 1,6-bisphosphate right here, we're going to cleave it right in half. On the left side, we're going to turn it into dihydroxyacetone phosphate. On the right side, it turns into glyceraldehyde 3-phosphate. So far, they're not identical yet, but it's very easy to convert this dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Eventually, eventually we will convert this into this. In the process, only glyceraldehyde 3-phosphate continues with glycolysis. The dihydroxyacetone phosphate will get converted to the glyceraldehyde 3-phosphate. Now you have 2 molecules of glyceraldehyde 3-phosphate, right? Now that glyceraldehyde 3-phosphate, now we're pretty good, right? Remember, the point is to split it in half, have each 3-carbon sugar look identical. Now we're pretty close, right, to having, almost identicality right here, right? So we produce 2 glyceraldehyde 3-phosphate. Now we're gonna start to put energetic bonds into it and then take energy out. So this glyceraldehyde 3-phosphate gets converted to 1,3-bisphosphoglycerate. In the process, we produce NADH, right? We had to put 2 ATPs in to produce these, 2 of them, right? So we've already put in 2 ATPs. Now the second half in glycolysis, we're going to derive energy from that. The very first energy forming step is a production of 1,3-bisphosphoglycerate from glyceraldehyde 3-phosphate, right? So you have 2 molecules, right? So you go through this reaction twice. The first time you produce 1 NADH. The second time you produce a second NADH. That's where we get the 2 NADHs, right? So NADHs are produced in the formation of 1,3-bisphosphoglycerate. Again, these reactions, I only want you to know for the quiz, right? From 1,3-bisphosphoglycerate, we're going to remove one of the phosphate groups. This is what it looks like before. You have 1 phosphate, 2 phosphate groups, right? As we remove one of the phosphate groups from this, well, that phosphate group is removed, we add it to ADP to form ATP. Again, right, we have 2 of these, right, substrates, 2 of these, right, glyceraldehyde 3-phosphates. We had 2 3-bisphosphoglycerates, and now we're going to produce 2 ATPs when we form 2 3-phosphoglycerate, right? Again, first half, we put in energy. Second half, we start taking the energy out of the reactions, right? Eventually, the 3-phosphoglycerates turns into 2-phosphoglycerate and then phosphoenolpyruvate, right? And then in the last step, we take phosphoenolpyruvate or PEP and turn it into pyruvic acid or pyruvate, right? In the process, we form 2 more ATPs. Again, I know we went through a lot of chemicals and the names are going to sound very similar, right? This is the reason why it's on the quiz, not on the test, right? Again, in terms of the test, I want you to think big picture stuff here. For the test, right, I want you to think that first step, right? That's it in terms of the reaction to glycolysis and the big picture. The big picture is we're trying to change glucose around so then when we split it in half, 3 carbon sugars on each side look similar, almost identical, and then we work on each of the 3 carbon sugars and produce pyruvate in the process. In the process of producing pyruvate, that last half, look at all the energy we produce, right? 4 ATPs, 2 NADHs, right? I say a lot, that's not a lot, right? So we can see 4 total ATPs are produced, but we have to put 2 in the beginning. So we only make 2 net ATPs. Now, NADHs will produce ATPs later on, but in order to produce it, you need oxygen, right? So what we get, that's the best product here, the most important one, that's going to be the most energy producing, is going to be the pyruvates. You can produce 2 of them. And we're going to see with each of the pyruvic acids, in the presence of oxygen, they go into the mitochondria, where each one will produce 15 ATPs each, right? Now, it doesn't produce it immediately. We're going to see each pyruvate is going to produce a lot of NADHs and 1 net ATP immediately, right? Eventually, though, those NADHs and FADH2s from pyruvate will eventually turn into 15 ATPs each. So that's a lot, right? So I'm not going to show you the animations. We will probably go through a little bit of this tomorrow. Again, right, this kind of gives you another look at this lactic acid fermentation. In the absence of oxygen, the sole purpose is to produce just enough ATP to survive, right? Just so you can, let's say you're in an area where there's not a lot of oxygen. You're able then, like let's say there's a fire and you have to get out. Well, in the process, if you breathe in that smoke, there's not oxygen in there. So your body will then need to use this lactic acid fermentation. What happens then? We'll produce just enough ATP. So then you can hopefully walk out of the situation. If you walk out, you take a big breath. Now we have oxygen available. That pyruvate then gets turned into acetyl coenzyme A in the mitochondria, and then we're going to start producing ATPs again, allowing us to live. But if you stay in that low oxygen environment, what happens? That reaction continues. All the glucose then gets used up, right? You form a lot of lactic acid and you form very little ATPs. And that's why we die when there's a lack of oxygen, is because you don't make enough ATPs to survive if there is no oxygen available. Right? Let's take a break before we go through the Krebs cycle. I know you guys are gonna, right? So let's take a 5-minute break and then we'll go through the Krebs cycle. Any questions, guys?
No.
Yeah, I guess not. Right? Again, right? Review this recording when I post it up. So then, right, I'm going to